Hello students, myself is Parminder Singh from Prab Kirpa classes. Our today's topic is the fundamental unit of life, part 2. In part 1, we have already learnt who discovered cells, what are the three basic components of the cell theory, what is the difference between multicellular and unicellular organisms, why cell is called the structural and functional unit of life. We have also learnt how division of labor is possible in multicellular organisms and why plasma membrane is called selectively permeable membrane and what happens if the plasma membrane ruptures or breaks down. We have also learnt about the process of diffusion and osmosis. We have also learnt the meaning of hypertonic, hypotonic and isotonic solution. In this course, we will discuss what is cell wall and whether the cell wall is found in animal cells. Next, what is meant by plasmolysis? Then, what is cytoplasm? Where is cytoplasm present? Then, what is meant by nucleolus? Then, if the organization of the cell is destroyed due to some chemical or physical influence, what will happen? Name the cell organs which are the sites of protein synthesis. Then, which cell organelle is known as the powerhouse of the cell? Then, what is meant by lysosomes? Whether lysosomes are present in plant cells? Why lysosomes are known as suicide bags of the cell? Then, name of the two organelles that contain their own genetic material. Why plastids are called the kitchen of the cell. Then, what are the various differences between animal cells and plant cells? Then, what are the various differences between prokaryotic and eukaryotic cells? Then, which type of cell division is required for growth and repair of the body, and which type of cell division is involved in formation of gametes? In this course, we will learn about cell wall, cytoplasm, nucleus, and ribosomes. First, cell wall. Plant cells, in addition to plasma membrane, have another rigid outer covering which is known as cell wall. This is picture of a plant cell in which you can see that this is the plasma membrane or cell membrane which is inside, and this is the outer covering. This is known as cell wall. So you can see from this picture that this plant cell has two coverings. One is plasma membrane and the outer covering to the plasma membrane is known as cell wall. Plasma membrane is found both in the case of plant cell as well as in the case of animal cell, whereas cell wall is found only in the case of plant cells. Cell wall is found in bacterial and fungal cells. The cell wall lies outside the plasma membrane or the cell membrane. The cell wall is absent in animal cells. What is the function of the cell wall? Cell wall provides structural support as well as protection to the cell. Now the question arises, from which material cell wall is made up of? The plant cell wall is mainly composed of cellulose. The presence of the cell wall enables the cells of plants, fungi, and bacteria to exist in hypotonic media without bursting. This is the picture of the hypertonic solution. This is the picture of hypertonic solution. If the plant cell is placed in hypertonic solution, you can see here that if this is the plant cell which has been placed in the hypertonic solution, what will happen? You can see that solutes are present in less quantity in the cell, whereas outside the cell, solutes are in dense quantity, more quantity. So what will happen? The water will leave from the cell to the outer, and only a few amount of water will come out. The net result will be that the more amount of water will leave the cell. And what will happen? Then the cell will shrink as the water will come out of the cell in more quantity. We call this contraction of the cell as plasmolysis. We have already learned about the hypotonic solution. That hypotonic solution is a solution in which the solutes are present in less quantity as compared to the another solution, that the solution inside the cell. When the plant cell has been placed in hypotonic solution, more water will come inside the cell than going out. So the net result will be that the plant cell will swell out, but the cell wall does not allow the cell to burst. It is only the cell wall which prevents the cell from bursting due to excessive water which has come inside the cell. Next, cytoplasm. You can see here that this is the cell, and the fluid which is present here all in the cell is cytoplasm, which is a fluid, gel like fluid inside the cell. So cytoplasm is composed of water, salts and various organic molecules. In the cytoplasm there are many specialized cell organelles. You can see here that this is the nucleus, this is the mitochondria, this is the Golgi body, these are the endoplasmic reticulum. There also lie ribosomes and lysosomes. These are mitochondria, Golgi body, ribosomes, lysosomes, endoplasmic reticulum. Each of these organelles perform a specific function for the cell. Cytoplasm is the ocean in which all the organelles float. You have seen that all these are the organelles, whether it is nucleus or mitochondria, Golgi body, these are endoplasmic reticulum, these are ribosomes. So all these are floating in the cytoplasm. Within the cytoplasm, materials move by diffusion. We have already learned about the process of diffusion in the part 1 of this course. Next, nucleus. In this picture you can see that this is the nucleus, this is the control center of the cell. It is an oval shaped body. This is the oval shaped body which is floating in the cytoplasm. We have already told you that this is all cytoplasm in which all the organelles are floating. It is not possible to see the nucleus with the naked eye. To see the nucleus, we have to take the help of a microscope. Nucleus consists of nuclear membrane, that is outer covering, nucleoplasm, nucleolus, chromosomes. Nucleus is separated. This is the nucleus. This is separated from the cytoplasm by a membrane which is called nucleus membrane. It means that there is an outer covering outside the nucleus which separates the nucleus from the cytoplasm. Nuclear membrane keeps the other organelles and components of the cell outside. There are small pores in this nuclear membrane that allow selected materials to go in and out of the nucleus. With the help of microscope of higher magnification, we can see a smaller spherical body inside the nucleus. You can see here that this is nucleolus. A small spherical body which is present inside the nucleus is called nucleolus. In addition to nucleus contains thread-like structures which are known as chromosomes. What is the purpose of chromosomes? These carry genes and help in inheritance or transfer of characters from parents to the offspring. The chromosomes are rod-shaped structures and are visible only when the cell is about to divide. Next, in case of prokaryotes, you can see here that this is the picture of a prokaryote. This is the picture of eukaryotes. In this picture, you can see that the nucleus area is well defined, whereas in the case of prokaryotes, nuclear region is not well defined. It is purely defined. In the case of prokaryotes, nuclear region is poorly defined due to absence of nuclear membrane and known as nucleoid. Whereas in the case of eukaryotes, nuclear region is well defined and surrounded by a nuclear membrane. Now the next question before us is, if the organization, the cell, is destroyed due to some physical or chemical influence, what will happen? We all know that as a building is made up of bricks, in the same way, all the living organisms are made up of small tiny units called cells. Cell is the structural and functional unit of all living organisms. If organization of the cell is destroyed due to some physical or chemical influence, all the activities performed by different parts of the cell, such as growth, reproduction of the cell, production of energy, removal of waste material from the cell, etc., will stop, and the cell will die, because there are many cell organelles which are present inside the cell which perform different type of functions, and if this organization of the cell is destroyed, then the cell will naturally die, because the functions performed by different cell organelles will not be performed. Now, what are ribosomes? Proteins are synthesized by ribosomes. Ribosomes are sites of protein synthesis. The ribosomes are very small and roughly spherical structures. You can see here that these are small spherical structures. These dots are small spherical structures. Ribosomes can be found floating within the cytoplasm, or they are attached to the rough endoplasmic reticulum. These are the rough endoplasmic reticulum, and ribosomes are attached to it. In this lecture, we will learn about mitochondria, Golgi apparatus, lysosomes, and plastids. First, mitochondria. In this picture, you can see that these are the mitochondria. Mitochondria all are small organelles inside the cells. You can see in this picture that the other organelles found inside the cell are nucleus, endoplasmic reticulum, Golgi bodies, ribosomes, etc. Mitochondria is present in both plant and animal cells. Mitochondria are known as the powerhouse of the cell. They are involved in releasing energy from food. Mitochondria stores and releases energy in the form of ATP. ATP is known as energy currency of the cell. Mitochondria uses oxygen and glucose to make OTP. Mitochondria move freely in the cytoplasm of the cell. You can say that mitochondria may move from here to here in the cytoplasm, from here to here, here to here. Mitochondria cannot survive outside of the cell. Mitochondria also make chemicals for other cell purposes such as breaking down waste products, responding to infections and injury, etc. Some organisms have a single large mitochondrion. Other organisms have thousands of mitochondria. Some organs like muscle, liver, kidney, brain contain higher number of mitochondria. There may be a question in the exam why mitochondria are strange cells. In what sense they are strange cells? So the answer will be the mitochondria are strange cells in the sense that they have their own DNA and ribosomes. So the mitochondria are able to make some of their own proteins. Now the next topic before us is Golgi apparatus. In this picture you can see here that this is the Golgi body or Golgi apparatus. Golgi apparatus is also known as Golgi complex, Golgi body, simply the Golgi. It is an organelle which is found in most eukaryotic cells. Golgi body was discovered in 1898 by Italian physician Camillo Golgi during an investigation of the nervous system. Now, what is the function of the Golgi body? Golgi body performs the function of modification, packaging, and transport of proteins and lipids, which are sent to different parts of the cell outside the cell. If there is no Golgi apparatus, then the packaging and transport of material to different parts of the cell and outside the cell will not happen. Others, then the other cells and organs in the body would not function properly without the products that the Golgi body normally sends. Now we will learn about lysosomes. Lysosomes is a membrane bound organelle that contains powerful digestive enzymes. In this picture you can see that these green color dots represent the lysosomes. Lysosomes are known to contain more than 60 different enzymes. This cell organelle is very acidic. Lysosomes are like the stomach of the cell. They digest dead or worn out or poorly working organelles. This is the function of the lysosomes. They also digest viruses or bacteria that enter the cell. They also remove the cell debris. What happens if the cell gets damaged beyond repair? If the cell gets damaged beyond repair, the lysosomes may burst and the digestive enzymes which are present in the lysosomes may eat up, digest other organelles of its own cell, which may lead to the death of the cell. So lysosomes are known as the suicide bags of the cell. So it means that if the cell is damaged from any part and it is not repairable by the internal system, then the lysosomes which are present inside the cell will eat all the other organelles which are present inside the cell, which will lead to the death of the cell. So due to this reason, the lysosomes are known as suicide bags of the cell. It is to be noted that lysosomes are absent in prokaryotic cells but present in all animal cells and in a few plant cells, that is fungi and yeast. Next, plastids. You may already know that plastids are not found in the case of animal cells. The plastids is a double membrane bound organelle. It is found in the cells of plants, algae, and other eukaryotic organisms. Like the mitochondria, the plastids also have their own DNA and ribosomes. The plastids provide color to the fruits and flowers. The plastids are involved in manufacture and storage of food. Plastids are called the kitchen of the cell. Why? Because plastids contain a pigment called chlorophyll, which plays an important role in the process of preparation of food by the plant cells called photosynthesis. It is because of the presence of the pigment called chlorophyll that the plastids are known as the kitchen of the cell. Because the chlorophyll plays an important role in photosynthesis. Now, there are three types of plastids. You can see in this picture chromoplast, chloroplast, and leucoplast. Leucoplasts are white colored or colorless plastids. They do not participate in photosynthesis. They are primarily organelles which store food such as starch, oils, and protein granules. Whereas chromoplasts are colored plastids other than chloroplast, usually containing yellow or orange color pigment. Now, chloroplasts, these are green colored plastids which help in the synthesis of food due to the presence of chlorophyll. Now, name the two organelles that contain their own genetic material. We have already learnt that mitochondria and the plastids are the two organelles that contain their own genetic material. Both these organelles have their own DNA and ribosomes. Now we will learn about endoplasmic reticulum, vacuoles, and cell division. First is endoplasmic reticulum. These two words consist of endoplasmic and reticulum. Endo stands for inside, plasmic stands for cytoplasm, and reticulum stands for network. It means that network inside cytoplasm. So collectively, the endoplasmic reticulum is a large network of membrane bound tubes and sheets. In this picture, you can see that these cream color and these stand for endoplasmic reticulum. The endoplasmic reticulum, in essence, is the transport system of the eukaryotic cell and has many other important functions such as protein folding. There are two types of endoplasmic reticulum. First is rough endoplasmic reticulum and the second is smooth endoplasmic reticulum. First we talk about rough endoplasmic reticulum. Rough endoplasmic reticulum looks rough under a microscope. Why it looks rough? Because it has ribosomes. You can see here that these red color dots represent the ribosomes. Because a rough endoplasmic reticulum has ribosomes are attached to the surface of the endoplasmic reticulum. The ribosomes are the sites of protein manufacture. The manufactured proteins are then sent to the various places in the cell depending on need using the endoplasmic reticulum. Now, the smooth endoplasmic reticulum. The smooth endoplasmic reticulum has no ribosomes. It appears smooth. It helps in the manufacture of lipids. The smooth endoplasmic reticulum also plays a major role in detoxifying many poisons and drugs in the liver cells of the vertebrates. Now we talk about vacuoles. Vacuole is a membrane bound organelle. These are small organelles which are found in animal cells, and whereas in the plant cell the vacuole is of much higher size as compared to the animal cell. Vacuole may or may not be present in the case of animal cell, whereas a vacuole of a higher size is present in the case of plant cell. Vacuole is a membrane bound organelle which is present in plant and fungal cells and some protist animal and bacterial cells. The vacuole is filled with water, sugar, minerals and proteins. As already told, animal cells may have no or many small vacuoles. In this picture, you can see that there are 3 vacuoles in the animal cell. Most mature plant cells have one large vacuole. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. What is the function of the vacuole? The main function of the vacuole is to handle waste products or store useful substances like water, proteins, etc. that the cell will need later on. In this picture, This is the vacuole which has been shown in the case of plant cell. In a single cell organism like amoeba, the food vacuole contains the food items that the amoeba has consumed. Now we discuss about cell division. The process by which the new cells are made is called cell division. Cell division is a process by which a mother cell divides into two or more daughter cells. When the cell divides, everything which is present inside the cell also divides, which means that the things like nucleus, chromosomes, and cytoplasm also divide with the cell. Cell division is the process which makes growth and reproduction possible for any organism. There are two main types of cell division. First is mitosis and second is meiosis. First, mitosis. The process of mitosis has been shown here. Mitosis is the process of cell division by which most of the cells divide for growth. In the process of mitosis, each cell called the mother cell divides to form two identical daughter cells. Since that this is the mother cell and it divides into two daughter cells. The daughter cells have the same number of chromosomes as mother cell. Mitosis helps in the growth and repair of tissues in organisms. Now, meiosis. It is a special type of cell division of germ cells in sexually reproducing organisms. In meiosis, a cell divides twice. You can see here that a cell has divided first this, second time this. A cell divides twice to produce four. You can see that 1, 2, 3, 4. A cell divides twice to produce four new cells. This was the one cell and it has divided two times to produce four cells. The new cells have half the number of chromosomes than that of the mother cells. Now we will do the recap of what we have learned so far. Cells have a cell wall outside the cell membrane. This cell wall is not present in the case of animal cells. Nucleus is the control center of the cell. Ribosomes are the sites of protein synthesis. Lysosomes are known as suicide bags of the cell because whenever the cell is damaged beyond repair, the lysosomes eat up all the other cell organelles which are present inside the cell. Mitochondria are known as the powerhouses of the cell because they produce ATP, energy currency of the cell. Prokaryotic cells have no membrane bound organelles. Cells in the organisms divide for what purpose? For the growth of the body, for replacing dead cells, and for forming gametes for reproduction.